The Kinexa technology provides information about binding affinity, kinetics, and concentration for unmodified molecules in solution. A kinetic exclusion assay is a method for quantifying the free fraction of one of the binding partners in a binding pair. This differs from most other technologies because they require one of the binding partners to be immobilized or modified to enable detection. Because of the high level of sensitivity, measurements of binding constants cover a wide range down to femtomolar. Some advantages of the Kinexa system include experiments that are easy to set up and run autonomously, versatility allowing for multiple assay formats, integrated software is simple and intuitive, yet powerful enough to analyze and diagnose more complex binding behavior, and the capability to measure unpurified materials, cellular expressed proteins, and small molecules without the risk of mass transport limitations or artifacts that may mask true binding constants. The instrument is equipped with a buffer reservoir, which is not limited to a particular type of buffer. This allows the user to determine the conditions needed for optimal binding. Samples are placed on the instrument, and each sample is measured autonomously when the experiment begins. Custom software allows the user to specify ideal sampling parameters and analysis type to match the assay format. Integrated one-click analysis uses exact solutions to the binding equation to calculate optimal binding constants that are highly reproducible. 95% confidence intervals are automatically generated. A confidence interval better represents this type of measurement than using standard deviation because the uncertainty does not follow a standard bell-shaped curve. In fact, most measurements are skewed to one side. Measurement of binding partners take place in the patented flow cell. Behind the optics cover, an optics system is used to amplify the signal generated from the binding partner being measured. Let's take a closer look at how the binding partners are measured for each sample. In solution, one of the binding partners is kept constant while the other binding partner is titrated. The titrated partner is also used to coat beads that are loaded into the flow cell prior to each measurement. Each sample contains binding partners that are both free and in a complex. The short contact time with the bead column does not allow for significant dissociation of the complex, so that only the free constant binding partner is captured. The amount captured is directly proportional to the free fraction of the molecule in solution. In order to quantify the captured molecule, a fluorescent label is introduced in a subsequent step. The fluorescent emission is then directly proportional to the amount of captured molecule. The kinetic exclusion assay is beneficial for any researcher looking to characterize binding pairs. Experiments are easy to set up and run autonomously. Software is simple, yet provides powerful single-click analysis with 95% confidence intervals, and the instrument saves secure data that cannot be modified to meet 21 CFR Part 11 compliance.